And welcome everybody to today's Drug Hunter Flash Talk. This is our first event of 2025, and I'm happy to be here with Adrian Gill from RevMed, and we're going to be talking about targeting the oncogenic state of RAS. Welcome to the show, Adrian. We're glad to have you today.
Thanks, Dennis. Great to be here.
Adrian is currently the Executive Vice President of Discovery Chemistry and the Head of Early Discovery at Revolution Medicines. And in his function there, Adrian has overseen the discovery and development of RevMed's clinical assets on a range of portfolio targets, including from the RAS, MAPK, and the PI3K mTOR pathways. And specific targets include the SHP2, SOS1, mTORC1, and the multiple RAS-on mutants that we will hear more about today. With over 28 years of experience in drug discovery and development, Adrian has held many senior roles in both large pharma companies as well as biotech environments, and has played a pivotal role in identifying numerous clinical candidates spanning really diverse disease areas. His professional journey includes contributions at Roche, Aztecs Therapeutics, and AstraZeneca, where at AstraZeneca he served as the head of the medicinal chemistry for the cardiovascular and metabolic disease group in Mölndal in Sweden. Adrian's extensive experience is reflected in over 30 publications in peer-reviewed journals, and his involvement as a named inventor on over 55 issued or pending small molecule patent applications. Adrian earned his PhD in organic chemistry from the University of Sussex in the UK, and his first class bachelor's degree in applied chemistry from the University of Salford in the UK. In addition to his role at RevMed, he serves as a scientific advisory board member for Curve Therapeutics and Expose Therapeutics as well, and he is an honorary professor of drug discovery at the Sussex University Drug Discovery Center in the UK, which is also his alma mater. Adrian, it's really a pleasure to have you here with us today.
Thanks, Dennis. Great to be here. Looking forward to this.
Awesome. And yeah, so before we get started, it's kind of a flash talk tradition, if you will, to ask some warm-up questions. So I want to ask you, Adrian, what inspired you to start a career in drug discovery?
Gosh, I guess I was always fascinated chemistry at high school in the UK, and I think for all of us who get into this business or anything that you really enjoy, you often have an inspirational figure in your life that introduces something to you. And mine was Mr. Redding, my chemistry teacher, really the comprehensive school where I studied my GCSEs back in the day. And he's a big Monty Python fan. And my friendship group and I at the time, we loved Monty Python, watching the films and the sketches as well. And he just found a really unique way of teaching, a tricky subject early on and keeping us entertained and sort of focused on the subject using the medium of Monty Python, which I thought was really quite inspirational. And that sort of set me on a path to go and do my degree at Salford University, which, I sort of really loved organic chemistry there as well. And I did 2 6-month industrial placements, one in a biotech and one in a large pharma. And that really helped me understand my career path in terms of what I wanted to do. And I work with some really smart people, very dedicated scientists, and it was just wonderful to see how I could use my sort of burgeoning synthetic skills to sort of directly improve people's lives with discovery of new medicine. So, and that's the potted history, if that's okay.
Yeah, that's amazing. Yeah, definitely a lot of inspiring figures, and we hope for great teachers in the field to really bring the chemistry to the next generation. Adrian, I think I might suspect the answer here from you already, but what's your favorite molecule and why?
As I was saying to you, as we chatted earlier, I don't really just have one. And maybe it's the sort of team sportsman and coach in me. As many of you who know me, I'm a big soccer fan, on Giants, and I've been sort of in the drug discovery business for nearly 30 years now, working across the UK, Europe, and East and West Coast of America across a range of different disease areas, and had the privilege of working with both the rule of 5 and beyond rule of 5 space. And I just really appreciate the tenacity and craft of the medicinal chemist and the drug hunter in every molecule that gets into the clinic or ultimately the market as well. But I know just how hard it is to get there as well. So I'm probably more about celebrating collective achievements here, of course, and what we learn from each other as a community. Yes, of course, it's a competitive business, but I think there's also an altruistic side to most of us as well. We want to see improvements in human health, and we love to see innovative small molecules progress as well. We've all lost family members. I lost my dad to heart disease, and my mum had colorectal cancer. So, I sort of think of myself as someone who celebrates our collective wins as a community, big and small, so we can learn from the hard knocks and try and move going forward. But having said that, as I said to you earlier, Dennis, I have to say, probably if you push me, it's a toss-up between actually two RevMed molecules. And I don't mean that it's because I drink my own Kool-Aid or anything like that. I do think they're pretty special, and that's RMC-6236 and RMC-9805 for they're essentially dogma-breaking profiles. Everyone thought 6236 was going to be horribly toxic, hitting wild type, and we've proven that differently, both preclinically and in the clinic as well. And again, 9805 has an aziridine moiety in there that many of us are taught is a toxiform and can't possibly go in a molecule. And here we are finding a way to fine-tune its drug-like properties and have a really unique safety profile as well. So, I'd say one of those two.
Yeah, that makes total sense, Adrian. I mean, it's always like picking your favorite child or something like that to pick your favorite molecule. So I do understand that's a hard one, but I think your choices are really very solid choices. And I'm excited to hear more about those today. What are you most excited in drug discovery today, like in general, Adrian?
I think probably in a nutshell, Dennis, I think molecular glue, fully exploiting the on-board 5 chemical space is sort of, it's a really big area that I think there's a lot of people working in, both big pharma and biotech and academics as well. I think that's a really exciting space that I think we've only just scratched the surface of, to be honest, and not just what we're practicing at RevMed, but all the TPD work that's out there as well. So that's what I'm excited to see where that goes next. I'm also a big believer in the AI machine learning components. I'm a healthy skeptic, so I know it's important, we need to use it, but at the same time, I don't follow the hype and believe it's going to change everything overnight and we're all going to be out of a job tomorrow. I think there's a lot of learning and progress that needs to be made hand in hand with that type of technology. So I'm very open-minded about it, but I think some caution along the way as well.
Sounds like two excellent choices, and we'll hear more about machine learning and AI and active learning as well in a few upcoming flash talks as well. So very excited to learn more about this. Yeah, Adrian, in your long-standing career, if you could give your younger self some career advice when you started your career, you kind of reflected a little bit on your career already when you did your bachelor's degree with the internships that you did. What kind of advice would you give your younger self and maybe our younger audience members that are just starting out their careers today?
Well, sort of thought-provoking question. Honestly, it's a lot of advice. I think it's probably what I would give myself, Dennis. It's probably too long for this flash talk, so I'll keep it to some of the main ones. One of the things I always say to my children and all new starters in all the companies I've worked with is be a sponge and act like every day is a school day. Ask lots of questions, seek out mentors and learn from them. It's really important in terms of building that community and finding your people to learn from and allow you to develop in your career. So don't be shy about building that network. I think Mark Merco said it very well on many occasions. Steal with pride. All good medicinal chemists have a mental Rolodex, as it were, of key building blocks or features of molecules that can solve various ADMET, structure-based design, potency, selectivity issues, etc. So that's really important to read widely and learn from people and just sort of collect those little gems so you can use them again. Be courageous, challenge the dogma. We've certainly done that at RevMed. I'd like to have done that more, perhaps at other points in my career as well. I've been very fortunate to work across a range of cultures and with different companies, so I would definitely encourage my younger self to do that again and perhaps do more of it as well. And probably the most important one is about hiring smart people, really people smarter than you, basically, and do everything you can to make them successful, because ultimately their success is your success. and drug discovery is a team sport. It's about the collective rather than the individual.
Awesome. Yeah, thank you, Adrian. And I understand you could probably go on and on and on with more advice. So we'll hopefully we'll have the opportunity to meet another day, and I can I can pull that out of you. All right. One more question I do have. What do you like most about Drug Hunter?
I think for me, there's a few things actually. There's the molecule of the month and the competition for molecule of the year as well. I really like because it's a very digestible sort of set of content and well curated as well. So there's a lot people to learn in a very short space of time there. The literature and patent summaries are great for various targets, what's going on, what's current. Conference updates, highlights is always great. And certainly the recent Flash Talk series, I've tried to connect as often as possible there. It's been really educational. Hopefully today won't disappoint either. So we'll see.
Sounds great. I'm sure you won't disappoint. Yeah, thank you for sharing that, Adrian. And yeah, before we get started with your talk, just wanted to share maybe one somewhat new feature that maybe not that many people know about that we have on Drug Hunter. And that's actually clinical highlights. So we do follow every month what is going on with key compounds in the clinic. So you can find that summary in every next month. So here in January 2025, we have the summary of key clinical updates from December 2024. And what that shows you is really key FDA approvals that happened in that month. So very famously, Vertex's, triple combination here got approved as a cystic fibrosis corrector with that really interesting looking Mickey Mouse type molecule here, vanzacaftor. Also, indication expansions here. Zepbound was now approved for obstructive sleep apnea. But we also talk about, rejections as well. We talk about NDA acceptances by the FDA as well. So you can see really a summary of what's going on and what's coming, what's hitting clinical trials soon. We talk about clinical trial initiations as well, positive clinical trial readouts, and what you can kind of learn from those with direct links to presentations and direct links here to clinicaltrial.gov as well. So you always know what's going on in the clinic with a lot more detail than you can find on clinicaltrials.gov with the structures of the molecules, a lot more context in which these results are really important. And then, of course, we also talk about negative clinical trial results. So that is, as Adrian pointed out, kind of a learning for the whole community of things that haven't worked out, and that will allow us to come up with better hypotheses going on in the future. And now, without further ado, I will hand it over to Adrian for sharing his presentation about targeting the oncogenic state of RAS with RAS-on tri-complex inhibitors. Adrian, the floor is yours.
Thank you, Dennis. Well, good morning, everyone. It's great to be here to give the first Flash Talk presentation of the new year. Let me start by thanking Gary, Dennis, and the Drug Hunter team, of course, for the invitation to give this presentation. Share with you some of the progress that we've been making at RevMed towards drugging the RAS oncogene. Slide, Dennis. All right, so our goal at RevMed is to bring sort of innovative targeted therapy options to patients with RAS-addicted cancers, which represent almost 30% of human cancers, by using a differentiated therapeutic strategy that's focused on the discovery of small molecule inhibitors that can selectively target RAS or RAS-on, as we like to call it. As many of you will be aware, RAS proteins are essential to maintaining normal cell growth and signaling. RAS itself is a small GTP-bound that essentially acts as a molecular switch and toggles between the off and the on forms. The off form being the GTP-bound state, and the on form being the GTP-bound state. And under normal circumstances, homeostasis is tightly regulated, modulating the population of RAS in the on state versus the off state, obviously to control cell growth. And in RAS-addicted cancers, of course, mutations in RAS shift this balance towards the on state, leading to increasing and uncontrolled cell growth. Mutations in RAS, of course, can be found in three main isoforms, K, H, and NRAS accordingly, and three hotspots, notably G12, G13, and Q61. The mutations in KRAS being by far the most common. These mutations are the main drivers for nearly a quarter of a million new cancer cases in the US alone. And I think even though we've known a lot about the epidemiology and biochemistry of various RAS mutated cancer for some time now, RAS itself has remained a pretty elusive therapeutic target until very recently. Move on to the next slide, Dennis. Thank you. All right, so the epidemiology of RAS-driven tumors is rather heterogeneous, both between and within tumor types. On the left-hand side of this slide, you can see the RAS driver mutations that are found with the mutational hotspots at G12, G13, and Q61. While mutations at glycine 12 account for about 80% of total numbers, only the small sort of 11% KRAS G12 superfraction addressed by currently approved agents, as far as that's expected, which means a large unmet medical need for multiple other RAS mutants are broadly distributed across several tumor types, and that's shown on the bar charts on the right-hand side. We move to the next slide, Dennis. So as I mentioned earlier, under normal conditions, RAS exists in this balance between the GTP-loaded off form or the GTP-loaded on form. And RAS proteins are activated in response to a variety of growth factor stimuli, which allows guanine nucleotide exchange factors, or GEFs, to exchange GTP with GTP, and therefore activating RAS. In the on state, RAS itself undergoes a sort of a conformational change, which is highlighted by the changes in the switch 1 and 2 region shown in blue and brown on the graphics here. And that allows RAS in its on form to now bind and activate effector proteins to promote cell division and survival. So when GTP is hydrolyzed to GDP, either by the GAP proteins, the GTPase activating proteins, or much more slowly through sort of intrinsic GTPase activity, the protein then reverts off into the off state and is unable to bind and activate effector proteins like RAF, for example. And oncogenic RAS mutations disrupt this balance by effectively disabling the GAP-stimulated GTP hydrolysis and increase the abundance of RAS-on protein that drives uncontrolled cell growth, of course. Now, as many of us know, RAS was thought to be undruggable due to the absence of obvious sort of binding site to binding pockets for small molecule inhibitors, and of course the picomolar affinity for both GDP and GTP as well. Yet about a decade ago, there was a major breakthrough by the Showcat lab and John Oster and colleagues using a cysteine tailoring strategy. We identified the induced binding pocket on GTP-bound KRAS G12C. And this led to the discovery of RMC-6291 that binds to and track RAS in the off state by forming a covalent bond with the oncogenic system as well. Obviously, we're all aware of subsequent efforts by many companies, particularly Amgen and Mirati, that have led to accelerated FDA approvals for cytosine RAS signaling treatment with KRAS G12C mutated non-small cell lung cancer, and of course, more recently full approval in colorectal cancer as well. But no doubt about it, was a huge leap forward for the drug discovery community, because not only did it validate direct targeting of RAS, of course, as a viable strategy for patients with RAS-mutant cancers, but also reinvigorated the sort of collective drug discovery efforts against RAS as a target. And as I'm sure many of you are aware, following on from the sort of initial advances, that many more companies have disclosed KRAS G12C off inhibitors, in addition actually to compounds that now target additional mutants such as KRAS G12C and KRAS inhibitors that target multiple KRAS mutants as well. But to the best of our knowledge, these inhibitors, like the first generation G12C inhibitors, all exhibit their highest affinity for the inactive state. So going forward, we felt that there's definitely a need for inhibitors that directly target on form of RAS, which is obviously responsible for effector activation should grow. And inhibitors that target the on state, we hope won't be as susceptible to mechanisms of escape from therapeutic pressure, but increase obviously the levels of RAS on through increase of receptor targeting kinase activation of RAS. But of course, direct targeting of RAS-on has been very challenging with conventional methods. And secondly, the featureless surface of the protein. The best analogy was provided by the Amgen scientist Vic C. and Brian Manman back in the day when they first disclosed the structures of Taurus. It's basically like a Death Star. It's completely smooth and featureless, no obvious pockets there to go after. And obviously with the picomolar affinity for GTP and GTP, as I mentioned earlier. So one approach to designing inhibitors that might target active RAS takes inspiration from the mechanism of action of natural products like cyclosporin and rapamycin, shown here, which behave essentially as molecular glues, and both of which are obviously approved drugs for use in humans as well. And as you may be aware, both these molecules first bind to an abundant cellular chaperone protein, in this case, the immunophilin CypA, cyclophilin A, and FKBP12, respectively. And the binding of the natural product ligand effectively sort of remodels the surface of the immunophilin and creates a binary complex that has high affinity for the target protein. And then upon binding to the target, sort of drives target protein inhibition. And some years ago, Professor Greg Bedine and the scientific team at Walk Drive Bio had the idea that there could be more examples like this in nature, and additionally that synthetic ligands based on those natural products examples could be used to redirect these immunophilin targets, or immunophilin ligands rather, to inhibit target proteins of therapeutic interest, including obviously active RAS. And then in the October of 2018, Revolution Medicines acquired Walk Drive Bio, and we've subsequently built upon their very early work and have since created a diverse portfolio of orally bioavailable beyond rule of 5 RAS-on inhibitors with, I feel, pretty unprecedented drug-like features, but you can all make your own minds up in that regard. And we'll talk about a few of them today later in the talk. We go on to the next slide, Dennis, thank you. So in this slide, you can see that our RAS-on inhibitors are small molecules that can enter a cell and reversibly bind to the abundant cellular chaperone cyclophilin A, or CypA, as we call it, remodeling its surface. And of course, the resultant binary complex that's formed has high affinity for RAS-on and forms a reversible tri-complex. We can also form a covalent interaction with the RAS-on target, forming a stable tri-complex, and that gains further selectivity and maximizes target suppression as well, of course. And it's important to note that in the absence of the inhibitor, cyclophilin, CypA, cyclophilin A itself actually has no affinity for binding to RAS, and conversely, the inhibitor has no affinity for RAS in the absence of cyclophilin A, which is a really key feature of our mechanism of action. And as you can see highlighted at the bottom of the slide, we've now declared 6 development candidates, both non-covalent and covalent mutant-selective inhibitors that target each of the 3 hotspots and mutations in RAS, namely G12, G13, and Q61. In addition, obviously, to a pretty groundbreaking RAS multi-inhibitor, RMC-6291, which is now to be known by its generic or international non-proprietary INN name, daraxonrasib. And I'll toggle between those 2 names throughout the talk. And that inhibits both mutant and wild-type variants of the 3 canonical isoforms of RAS. And we're also really proud to note that the generic names for the drug candidates actually are all centered on the new phrase "on-rasib" that alludes really to the innovative mechanism of action for this new class of compounds. And 3 of the compounds highlighted by the asterisk are currently undergoing clinical evaluation, with daraxonrasib now in phase 3 trials for PDAC, pancreatic ductal adenocarcinoma as well. But I won't be touching on the clinical data today. I'll focus more on their discovery and preclinical profiles. If we go on to the next slide, Dennis, thank you. So there are unique properties of immunophilin chaperone proteins that really make formation of a tri-complex possible. Among them being obviously the high degree of plasticity and a large binding surface area. And the ability of CypA to adopt different conformations upon binding with different ligands enables us to sort of design and construct compounds that promote new partnerships with different targets. If you just click one more, Dennis, then you can see in the example here a tri-complex inhibitor, or TCI, as we like to call them, binds to CypA and remodels the surface of CypA to form a binary complex with a high affinity for the on form of RAS. Obviously, then using structure-guided techniques, we've been able to optimize these non-covalent interactions to build more potency and more, more drug-like properties, and actually, where applicable, also develop covalent interactions as well to identify TCIs with a diverse spectrum of properties. We've gone to the next slide, Dennis. Thank you. So this slide provides like a high-level overview, if you like, of our TCI platform. On the left, we've got 6291 or elironrasib, our G12C selective covalent inhibitor. On the right-hand side is daraxonrasib, which is obviously our RAS multi-inhibitor. And these two compounds essentially sort of represent the current bookends of the TCI platform that gives us broad access to potent and selective beyond rule of 5 RAS(ON) inhibitors with diverse profiles and retain good oral bioavailability and drug-like properties as well. Using structure-based design, as I said previously, we'll be able to optimize various features of a TCI to dial in various properties such as cyclophilin A affinity to enhance potency and increase non-covalent binding to give TCI formation and increase cell potency, of course, and also build in specific covalent or non-covalent interactions to drive selectivity for any given mutant as well. And this includes sort of the warhead and linker optimization in the case of RAS mutants with amino acid residues that are susceptible to covalent modification. And to date, I did the numbers just before the holiday break. We've made close to 40,000 beyond rule of 5 tri-complex inhibitors now and sold over 1,500 tri-complex crystal structures across the RAS isoforms and mutational hotspots. So it's been a pretty productive sort of 6+ year period, that's for sure. If we go on to the next slide, Dennis. So this slide sort of outlines the mechanism by which the tri-complex inhibits RAS(ON) and abrogates the oncogenic signaling cascade. So on the left-hand side is a sort of surface representation of a high-resolution tri-complex crystal structure with RMC-6291, which is a preclinical covalent G12C inhibitor, sort of tall compound that's representative of the clinical candidate elironrasib with the on-state of KRAS G12C and CypA, of course. And formation of the tri-complex rapidly inactivates RAS signaling by sterically occluding binding and preventing activation of downstream RAS pathway effectors such as RAS proteins, of course. And experimental evidence is shown here, looking at a biochemical cell-free assay, which is the sort of the middle left of the slide, and the impact of an inhibitor on the interaction between G12C and wild-type RAS, and of course the RAF binding domain, the RBD, monitored using TR-FRET. And we can see that the compound inhibits mutant but not wild-type RAS interactions in a concentration-dependent manner. And obviously, in the absence of CypA, there's no effect, consistent with the need for tri-complex formation and our mechanism of action, of course. In the middle graph is we're looking at the kinetics of disruption of the RAS-RAF interaction in a cellular context using nanobrex. And we see, again, immediate disruption of the signaling consistent with immediate engagement of the RAS(ON) protein as well. And then lastly, on the right-hand side, you can see the cellular consequence shown here for inhibition of phospho work. If we go on to the next slide, Dennis, thank you. And then sort of some of the key differentiating features of RAS(ON) inhibitors versus the inactive state selective inhibitors are sort of shown here. In the top left, which is the cellular RAS-RAF disruption assay, you can see the ON inhibitor immediately inhibits signal again versus the sort of two off inhibitors used here, adagrasib and sotorasib, which have much slower kinetics, which really reflects the rate of intrinsic hydrolysis of G12C, and it takes time to generate the GTP-bound form, which is obviously what the off inhibitors bind to. A similar profile is seen in vivo on the right-hand side. Here we're monitoring G12C target engagement in tumor after a single dose, and the ON inhibitor again rapidly engages target, and at higher doses this is sustained out to 24 hours, of course. And as I think I described earlier, activation of RTKs and GETs by either non-genomic or genomic mechanisms will drive the equilibrium for G12C from the off to the on form, or the on state. And we can model this in vitro and examine impacts of on versus off inhibitors as well.
So in the bottom left, using a phospho-ERK assay in the presence of increased RTK activation for a G12C on inhibitor in green versus a G12C off inhibitor in blue, as you can see, upon addition of exogenous growth factors to activate RTKs, only the off inhibitor loses potency. And in the bottom right, we've engineered a G12C mutant cell line to overexpress RTKs or express RTKs with activating mutations in the kinase domain. And again, we see a similar shift in potency for the off inhibitors, but not the on inhibitors. And we feel that collectively, these data highlight the advantages of on versus off inhibitors, and think it gives pretty compelling evidence that there's definitely a reduced susceptibility to adaptive feedback mechanisms that activate RTK signaling for the on inhibitor class. If we go on to the next slide, Dennis, thank you. So I think let's move on to the specific compounds in the late stage in the clinic now, and I'd like to share a little bit more about our investigational agent, daraxonrasib, or RMC-6291, our RAS multi-inhibitor. As you can see in the middle of the slide here, we can see a representation of a high resolution crystal structure of daraxonrasib with KRAS G12V in this case. Daraxonrasib does not really discriminate between HN and KRAS, as the sort of divergent residues between these isoforms are pretty distal from the protein-protein interaction interface in the PPI interface in the tri-complex, which is highlighted in blue. In addition, obviously, this compound occupies an induced binding pocket between CypA and mutant RAS, similar to elironrasib as well, except that actually it leaves a groove containing the common oncogenic mutant residues at G12, G13, and Q61 unoccupied. And this really provides the structural basis for daraxonrasib to bind multiple RAS variants. And we've got crystal structure data supporting that daraxonrasib exhibits a consistent binding mode across all of the KRAS G12X mutants tested as well. And on the right-hand side, you can see the biochemical data demonstrating sort of potent disruption of RAS-RAF at wild type and at various hotspot mutations as well, with potencies generally in the range of 3 to 10-fold of each other as well. We've gone to the next slide, Dennis, thank you. We carried out also a large cell panel screen for cell viability in response to daraxonrasib treatment in collaboration with the PRISM group at the Broad and also the Dana-Farber Cancer Institute as well. The volcano plot on the left shows one depiction of the results from the 870 cell line survey that clearly finds RAS mutation. Obviously, it's the strongest genetic determinant of sensitivity in the screen. And as expected, the BRAF mutation correlates with a lack of response, likely driven by sort of V600 mutations that don't rely on upstream signaling, of course. In the middle panel, activity of daraxonrasib against different KRAS mutants is shown, and a lower AUC indicating a greater inhibition of proliferation. As you can see with cell lines with KRAS mutations at codon 12, they stand out as being particularly sensitive, although the compound is also active against Q61 and has some activity against cell lines with wild-type RAS as well. And we think the increased sensitivity of cell lines with mutation at G12 rather, is not actually a function of properties of the compound itself, but actually rather reflects something about susceptibility of G12 bearing lines to inhibition of RAS. And as can be seen by the DETMAP genetic knockout data on the right-hand panel, showing a striking similarity profile to RAS multi, which gives us pretty much significant confidence in the on-target activity of our RAS multi. We go on to the next slide, Dennis, thank you. So translating these findings in vivo, we carried out a mouse clinical trial with daraxonrasib across a large panel of RAS-driven xenograft models of KRAS G12 mutant tumors in vivo. Shown here are the results from the KRAS G12X models across 3 major cancer types in which these mutations are drivers at a well-tolerated dose of daraxonrasib. And the color code represents a particular mutation in the waterfall plot, obviously captures representative responses from each of these models as well. And as you can see, we've observed some pretty remarkable responses across the majority of non-small cell lung cancer and PDAC models. And as expected, colorectal cancer responses are a little more diverse, consistent with the fact that multiple drivers are often found in this disease and co-mutations like PIK3CA, for example. And we're currently mining the genomic data across these models to identify determinants of response and resistance, of course, as well. On the right-hand side, we can see the durability of the responses in the models depicted in the waterfall plot, and obviously the green line represents individual tumors from approximately 50 models as well. And you can see compared to controls in black, daraxonrasib treatment results in significant and durable disease control out to 90 days. And with such a profile, we just felt compelled to advance daraxonrasib into clinical development. And we did that in mid-2022 with a goal, obviously, to translate the attractive preclinical profile into the clinic. Next slide, please, Dennis. Thank you. So switching gears now to focus on KRAS G12D, which actually has the largest population of any of the oncogenic RAS mutants, with over 61,000 new patients a year in the US alone. The primary histologies of these patients are colorectal, pancreatic, and non-small cell lung cancer. And all of the patients with PDAC that were sequenced in the Foundation Medicine's database, nearly 40% actually had a KRAS G12D mutation. Nearly 15% of colorectal cancer patients also had a KRAS G12D mutation, and with 4% of patients having a G12D mutation in non-small cell lung cancer as well. And RevMed's in a very fortunate position that we actually have two clinical stage approaches towards advancing treatment for these patients. The first, of course, is our RAS-on multi-selective inhibitor, daraxonrasib, that also is potent against G12D. And our second approach, that's going to be the focus of the next part of this talk, is our RAS-on G12D selective covalent inhibitor, RMC-9805, or zoldonrasib. And we believe these two approaches are complementary. They offer differentiated target profiles and may even enable sort of distinct standard of care combinations, as well as the possibility of a doublet combination with the two RAS-on inhibitors as well. And we think it collectively provides the potential for innovative new therapeutic approaches for patients with KRAS G12D mutant cancers. Go on to the next slide, please, Dennis. Thank you. So RMC-9805 or zoldonrasib is a pretty remarkable compound, as it is, to the best of our knowledge anyway, the world's first selective covalent inhibitor of aspartate G12 in mutant KRAS. And as recently stated in the American Society of Clinical Oncology Education book, and I'll read this, chemical leverage of aspartic acid by covalent G12D inhibitors will likely be impossible. I think the statement pretty much clearly underscores how challenging targeting aspartic acids covalently can be. And unlike targeting cysteine, for example, approaches to targeting aspartic acids have pretty limited precedent due to a number of factors, including things like significantly lower reactivity and nucleophilicity, of course, of the aspartic acid relative to cysteines. The warheads, as I'm sure many of you are aware, documented in the literature, overcome this by being highly reactive and unfortunately unstable, both properties of which reduce their usefulness in drugs, but provide some utility as chemical biology tools, of course. In addition, obviously, to the chemical characteristics of the aspartic acid warheads, selectivity is also a larger sort of issue for us, because there's like 5x greater number of acidic residues on the surfaces of proteins relative to cysteine as well. So with this in mind, we sort of had to intentionally reduce the reactivity of any particular warhead to make these compounds more drug-like, which sort of further compounds the challenge of achieving sufficient on-target reactivity with an aspartic acid at position 12. But nature, once again, however, has figured out how to do this in various enzymes by exploiting a large surface area with many non-covalent interactions to precisely position an aspartic acid residue in a privileged environment to form a covalent adduct with normally unreactive substrates, while also sort of precisely positioning the substrate and changing the local sort of electrostatics and excluding bulk water. If we go on to the next slide, Dennis. Thank you. So as for natural enzymes, the tri-complex can also form an extensive buried surface area and multiple non-covalent interactions, protein-protein and protein-ligand contacts, so that we can leverage these unique features to our advantage to really ensure sufficient reactivity and specificity for the substrate, as sort of illustrated hopefully in this close-up of the CypA, KRAS G12D, PPI bound with a RevMed compound that's shown in blue. The tri-complex sort of binding site can then be engineered to precisely position the warhead and via the linker, of course, and the 12D residue in optimal orientation for reactivity, while also modulating the local electrostatic environment and excluding bulk water to drive sufficient on-target reactivity. And as I mentioned earlier, most of the known literature on carboxylate sort of reactive functional groups documents their very poor drug-like properties, including intrinsic chemical stability. And early in our program, we wanted to make sure we screened these things out and make sure that we could sort of have that balance between intrinsic reactivity with drug-like properties. And really for us, the optimal warhead is one that has that low intrinsic reactivity to reduce the potential for off-target effects, but also has the opportunity to have sufficient on-target reactivity in the tri-complex environment that secures covalent cross-linking as well. And through our early screening efforts, really, aziridines emerged as the favored sort of starting point to achieve this optimal profile. And if we go on to the next slide, Dennis, thank you. So diving into the MedChem campaign then, non-covalent compound 1, as shown here, was an early lead in our RAS multi-program and one we published on last year in Science and Nature accordingly. And it afforded a really good trajectory towards the aspartic acid in KRAS G12D, to which we added our aziridine warhead, resulting in compound 2. And this really sort of represents our first G12D site-selective covalent compound with about 34% cross-linking over 6 hours in a biochemical assay, with a concomitant sort of increase in cellular potency in the ASPC1 cell line in both the phosphor work and CTG assays as well. And as you can see on the right-hand side, an X-ray crystal of this compound with KRAS G12D showed a clear and unambiguous continuous density indicating a covalent bond. We go on to the next slide, Dennis, thank you. In the X-ray crystal structure, we noticed that even in the post-cross-link sort of configuration, there were 2 sort of distinct conformations as shown in the top right-hand side in blue and grey respectively. And keeping in mind the importance of positioning the warhead, we really looked to try and rigidify the linker and enhance cross-linking further. And really, through incorporation of a 4-4 spiral motif, we're able to increase the cross-linking rate to 71% over 6 hours. And then using sort of molecular dynamics to sort of explore the pre-reacted conformational populations, you can see in the graph on the lower right, the warhead from compound 3, shown in green, spends a little more time in close proximity to D12 than the matched pair shown in blue with a flexible linker. We wanted to continue to build on this. We go on to the next slide, Dennis. So after much analoguing around the warhead, we also found that incorporation of a cyclopropyl moiety onto the beta position of the aziridine led to a consistent increase in covalently cross-linking the aspartic acid residue, and that allowed us to move cross-linking from 71% to 89% for compound 4. And this increase obviously was consistent across a range of matched pairs, which is sort of shown in the graph on the right-hand side as well. And really this increase in cross-linking can be down to a number of reasons. I don't think we've actually nailed it's precisely why, but we've got a number of different theories here, which is really around sort of modulation of the aziridine PKA, potential hyperconjugation effects, of course, with the sort of pseudo aromaticity of the cyclopropyl moiety as well, or it could be just restriction of conformational flexibility of the warhead as well, or a number of those factors combined together. But also, unfortunately, the substitution also introduced instability in simulated gastric fluid as well. So, and as we've reported in the past, having a phenol in the macrocycles certainly was not ideal for oral bioavailability as well. So if we go to the next slide, Dennis, just really cutting a long story short, and obviously in the interest of time today, we did a huge amount more optimization of the lead series, as you can see here, and that ultimately led to RMC-9805, or zoldonrasib, it's now called, an orally bioavailable beyond rule of 5 covalent RAS-on G12D selective inhibitor. Biochemical cross-linking rate is over 86% at 6 hours, low double-digit phosphor work and CTG potency in KRAS G12D mutant cancer cell lines, and respectable solubility for a molecule of its size as well. the SGF, GSH, and whole blood stability, again, very respectable, over 120 minutes. And the average oral bioavailability across preclinical species is over 30% as well, with moderate clearance. We also feel it has a low risk profile with respect to the safety panel and cystinome screens that we've also looked at as well. On to the next slide. Thank you, Dennis. So zoldonrasib itself and its unique sort of cyclopropyl aziridine, obviously highly selective for cross-linking RAS G12D, and I'll walk you through some of that data using various orthogonal methods on this slide as well. So we start on the upper left side. Cross-linking to RAS is obviously apparent as an upward band shift that you can see here, and is observed only for KRAS G12D and not for wild type 13D, 12C, or 13C as well. And we're also able to cross-link other G12 RAS isoforms, exemplified here. We have NRAS G12D as well, and obviously that's sort of anticipated given the sequence identity in binding site across HN and KRAS, of course, as well. We further explored the selectivity of zoldonrasib, profiling sort of biochemical cross-linking to very closely related mutants as well, and even with a modest change of the nucleophile in the 12 position from aspartic acid to cysteine or even to glutamic acid, it significantly reduces cross-linking. And again, similarly, shifting the aspartic acid residue from 12 to residue 13 completely abrogated cross-linking as well. And then consistent with the tri-complex mechanism and selectivity for RAS-on, one can see that not only is cyclophilin A necessary for cross-linking, but also no cross-linking is observed to the GTP-bound or the off state of KRAS G12D as well. And then lastly, on the right-hand side, we generated a clip probe to broadly interrogate sort of off-target reactivity. And obviously in the composition experiment in SPC1 cells, zoldonrasib pre-treatment led to a significant decrease in enrichment only for KRAS G12D, which is consistent with the selective binding here as well. We go on to the next slide, Dennis. Thank you. So on this one is the X-ray crystal structure evidence for covalency, with the left hand side graphic showing the clear covalent bond to 12D, and then on the right hand side you can see the many protein-protein non-covalent interactions that allow for this sort of enzymatic reaction to essentially occur. Go on to the next slide, Dennis. So using a sort of a similar paradigm to the one I showed earlier for elironrasib and daraxonrasib, we initiated a mouse clinical trial for zoldonrasib as well, and we saw obviously significant inhibition of tumor growth across multiple CDX and PDX models here, across a range of histotypes as well. And really based on this attractive preclinical profile, we decided to advance RMC-9805 or zoldonrasib into the clinic. We go on to the next slide, Dennis. I'll skip this, I think, in interest of time. I think people can sort of read through that at the leisure, or watch it again on the video. So if we go on to slide 25, thank you. We're obviously very encouraged by the preclinical profiles and the early clinical data for our RAS-on inhibitors, and we appreciate that both intrinsic actually and acquired resistance to targeted therapies is very common. Therefore, a core element of our strategy is to advance sort of optimal combination regimens of mutant-selective and a RAS multi inhibitor as well. And these agents have complementary profiles and obviously can be deployed as both monotherapy or combination against RAS-addicted cancers. And we believe that actually the doublet of RAS-on inhibitors offers a really unique opportunity to maximally suppress the mutant allele, while at the same time hopefully mitigate some of the diverse mechanisms of tumor escape. Go on to the next slide, Dennis, thank you. So by way of example here, we've got some of the combo data in G12C non-small cell lung cancer models, where we selected a subset of these models that exhibit sort of suboptimal responses to single agent G12C on inhibition, shown in blue, and including 5 that were identified as being resistant to elironrasib. RAS multi can drive a deeper response in some of these models, but it's really clear that the optimal response is seen with the combination, and we now see regression in the majority of these models. And when you look actually at the longer term dosing out to 90 days, the benefit of the combination is even more apparent, with increased durability of response, and something we consider to be particularly important for translation into the clinic. We go to the next slide, Dennis, thank you. So in addition to sort of defining optimal combination strategies to target sort of tumor intrinsic mechanism, we're also exploring sort of preferred strategies for combining our RAS inhibitors with IO therapy as well. And it's sort of well established now that mutant RAS is a key driver of immune escape through multiple mechanisms. On the one hand, activation of RAS drives an sort of immune evasive tumor microenvironment, modulating the tumor cytokine milieu to promote sort of infiltration or of immunosuppressive immune cells such as macrophages and neutrophils, while at the same time inhibiting the infiltration of T cells as well. And on the other hand, you've got the activation of RAS that disables tumor recognition by the immune system, that leading to sort of downregulation of antigen presentation machinery and obviously upregulation of immune checkpoints. If we go on to the next slide, Dennis. Thank you. So again, I don't have a lot of time to share all the data with you today, but we and many others have shown already that inhibition of mutant RAS with direct RAS inhibitors off and on, but particularly on in our case, can reverse many of these changes. And collectively, we think RAS inhibitors promote changes in the tumor and the tumor microenvironment that really are very favorable for combination with immune targeted therapies. And we recently shared data, early clinical combination safety data with daraxonrasib and elironrasib with pembrolizumab at the end of last year at the ENA meeting in Barcelona. So we've gone to slide 29. Thanks, Dennis. So really getting close to the end now, I promise. I wanted to close out by highlighting our pipeline from our most recent sort of corporate update at JP Morgan that really highlights our three lead investigational agents, daraxonrasib, elironrasib, and zoldonrasib, with the focus on their ongoing monotherapy and combination sort of clinical evaluation. Plus, of course, our additional RAS(ON) inhibitor assets, namely RMC-5127, which is our G12V selective molecule, RMC-0708, Q61H selective molecule, and also RMC-8839, our G13C selective inhibitor as well. We've now had pretty substantial clinical experience with our investigational agents. We've had more than 1,000 patients participating in a clinical trial with one or more of these agents. And really our development vision is for these assets is sort of to maximize the clinical impact across tumor types and lines of therapy through both single agent approaches and, of course, combination strategies as well. So we'll just go on to the last slide. So it just sort of remains for me to say, a huge thank you to all of my R&D colleagues for their sort of tireless commitment to patients, and our many research and clinical collaborators as well, of course, and of course all the patients and their families as well that have been involved in our clinical trials. And really for further reading in the RAS drug discovery space, a shameless plug for this very fine book that Professor Kaivan Shokat and I have recently authored that really charts the last 40 years of RAS drug discovery and what we feel the future holds in the field, and so, available online and from all big bookstores as well. But thank you, everyone. Thank you again for joining this flash talk. Thanks to Dennis and the Drug Hunter team as well, and for the invitation. I'm very happy to take any questions that people may have.
All right. People are already really excited, and people are already asking a lot of questions. So let's launch right into it. So we have a question here asking a wonderful talk. What was the reason to choose cyclophilin A as the binding protein partner to RAS?
Yeah, that's a great question. That was really sort of legacy sort of information from the Warp Drive Bio group, and like I said, they really came up with this concept and the basic research, and did a lot of the early work in understanding what are the best sort of chaperone protein presenter proteins to use. And cyclophilin A, FKBP12, the two sort of most prevalent ones for usage, and they did a lot of early screening back in the day. There seems to be a really unique complementarity between cyclophilin A and the RAS protein, and that was what their early research was built on, and we carried that on and then re-engineered their early ligands into the drug-like molecules that we see today.
Yeah, thank you. And then Anne Tran is wondering, you mentioned the important roles of cyclophilin A and tri-complex affinity, but she's wondering what you can comment on, if you could comment also on the role of cooperativity in driving the tri-complex formation.
Yeah, no, it's really important. It's a big protein-protein interface, and we can sort of direct that with respect to changes to the small molecule ligand as well. And as I said, we can reprogram the interface of the binary complex to then target the protein of interest as well. And we've learned a huge amount over this last sort of 6+ years since we acquired Warp Drive Bio in terms of how to sort of re-engineer that, not just for CypA, but for other proteins as well. And it's always the balance of the affinity for the presenter protein and other components as well. And we're starting to share more about that in publications as well, and conference presentations going forward.
Awesome. Yeah, cool. We're definitely looking forward to those.
Mhm.
And then Matt Hess is wondering, can you comment on the importance of the inhibition of wild type signaling in 6236 treatment?
Yeah, I mean, I think it's a large component of the efficacy profile of 6236, because obviously if you hit the driver mutant, then there's either upregulation of that mutant as well. So you've got to keep thinking about the dosage, as it were, to directly hit that mutant allele. But of course, wild type plays a component as well, and that's also amplified and upregulated to some extent. So hitting that immediately switches off an opportunity for the tumor to try and escape, and dampens down. I think the other sort of mutational hotspots, clones that might be available to then sort of grow out and have that acquired resistance. And I think that's why we're seeing the really encouraging durability profile of 6236, that we hit not just the driver mutational mutations, but also the wild type component as well.
Awesome. Yeah, and then we have a question from Darren here. Contemplating other undruggable targets, are there structural characteristics of the proteins that make them good targets for cyclophilin A glues?
Simple answer, yes, and more to come in the future.
Love that. John Spencer says, brilliant talk, by the way. And he also has a very specific question here. I saw a nice indole trifluoroethyl group in one of your molecules. We developed a carbazole trifluoroethyl for a multipolar interaction in the P53 Y220C, and PMV have used a similar strategy in their clinical trial for the P53 reactivating drug. Is your moiety of that indole also targeting cysteine? Does this also does this also target the backbone peptides, as was surprisingly seen in a publication in 2016? From
great question, John, and hi, John. We've not spoken for a little while, so we should get back in touch in the new year, that's for sure. Great question. It actually doesn't. It just fills a lipophilic pocket and space between cyclophilin and RAS as well. And actually, we chose that moiety, as you'll see from other examples in 6236, the daraxonrasib and the leronrasib. They actually just have the ethyl group. We introduced the trifluoro group for 9805 zoldonrasib, just from the metabolic stability point of view. And it also gave us a little bit more potency by space filling that region a little bit more, but no other interactions otherwise.
Sounds good. Yeah, cool. Thanks for sharing that. And Chris Burke then is wondering, impressive that the final molecule contains only 2 hydrogen bond donors. How important is that for oral bioavailability?
Very. No, I think it's a great question. And it's one of those things that, we've all got our unique sort of rules and guidelines, haven't we, in terms of what makes these large molecules sort of orally bioavailable. And, we realize that, controlling rotatable bonds, hydrogen bond donors, lipophilicity, a range of factors for unique sort of RevMed rules, if you like, have sort of come together over the years. And that's why we've got 6 orally bioavailable molecules that are somewhere between 800 and 1200 molecular weight. So they're pretty heroic beasts of compounds. But across preclinical species, we've got some really favorable DMPK profiles there. So yeah, definitely an important property, but there are other sort of layers to the onion, as you might imagine, as well, of course.
Totally. Yeah, no, I can't wait for the beyond rule of 5 molecule rules to be orally bioavailable. I think a lot of people have published on similar things. Like we have like an article about that for the Werner helicase inhibitor for, that Novartis has brought. So having more, and then of course there's a lot of academic publications on chameleonicity. So it is really super cool, and definitely looking forward to the RevMed rules. We're a little bit over time, so I think let's close it. There's so many more questions, Adrian. So I think that talks about how brilliant this talk was and how really people are very excited about those strategies. But let's close it out with one last question here. Can you talk a little bit more on the potential of using macrocycles to amplify and generalize the application of such method? And is RevMed involved in doing that?
I'm not sure I understand the question, Dennis. So how do you mean going beyond RAS or just the beyond all the five chemical space?
Yeah, I think again, this is a question Lian Tang asked. I think it's asking more like in terms of macrocycles, maybe how important is that, and
Yeah.
can that be? I mean, maybe a similar question of what we already discussed, and you already answered yes, more to come in the future.
Yeah.
Can that concept of macrocycles, I guess, be used in a broader?
For sure. No, I think so. I mean, we obviously have a unique platform that has this binary complex, so it involves the larger surface area of a macrocycle with also a protein on top as well. So you get even more surface area that allows us to create some unique binding events to otherwise unrelated sort of target proteins as well. So sure, there's more for us to explore and learn. But I think that the macrocyclic field, it's been natural products and peptidic macrocycles out there for many years. People are working in that space, like the Orbis team in Denmark. We've got Curve Therapeutics in the UK as well. They've all got their unique flavors of building these sort of macrocycles or microcycles in some cases that try and confer more oral bioavailability properties into those chemistries that then allows them to go fishing for a range of targets beyond the oncology space, of course, as well. And actually without a binary complex in terms of a tri-complex sort of mechanism of action like we're going after. So I think it's a super exciting space. As I answered your question at the beginning about what am I excited about in the area, I think molecular glues, all things beyond all the five, I think we're only just scratching the surface here. We're going to see a lot more innovation going forward.
Awesome. Yeah, thank you so much, Adrian. Yeah, thank you for also sticking around a few extra minutes to answer these questions. Again, there are many more questions that we don't have time to answer today. So apologies for that. We may be able to get back to you in writing. Adrian, I want to thank you for a phenomenal talk. I want to thank our audience today for asking these very insightful questions. And we will be back on February 6th with Pat Walters talking about active learning and drug discovery. So be sure to dial in then. And for everybody, a wonderful rest of the week. And again, thanks, Adrian, for a phenomenal talk.
My pleasure.
All right. Cheers.